All right, we will jump right in. Hello, everyone. Let me share screen here. Okay, hello, everyone. This is the Symbulation tutorial. Hopefully, you are aware of where you are. So for the next hour and a half, we'll be talking about and getting some hands-on time with the platform Symbulation. I am joined today by several people that are helping organize things, so we will do introductions shortly. I'm Anya Vostinar. Yes, hopefully you see that. Before we jump into like the schedule and stuff, there are some things that will be helpful to download if you don't already have them. And so I think Piper's going to put the link to this in the chat. You got it, Piper? Perfect, awesome. At the Read the Doc Symbulation, there is an advanced prep thing that has links to a couple of different things to download that will be good to start doing while you are listening to us. Hopefully, some of it you might already have. Okay, so while we're doing that. The kind of schedule for today, first we'll be doing both intro of who we are and intro of the software. And then the first hands-on will be about 15 minutes. We'll take a break. And then we'll dive more deeply into the code structure. So like how the code actually works and like which things might be places that you'd want to make changes if you were wanting to do so. And then a chance to actually try that stuff out and wrap up quick. So it's a quick session, just trying to kind of introduce things, but then also a chance to try things out while you have a whole bunch of people here who are happy to help and debug and try and figure out what's going on. Okay, so as promised, intro. So I'm Anya Vostinar. I am a assistant professor of computer science at Carleton College, and I created and continue to maintain the Symbulation platform. I think that Emily is not here today. Emily is an assistant professor at Michigan State University and a major contributor to Symbulation. And then I will let Kira introduce.
Hi, I'm Kiara Johnson. I'm a rising junior at Carleton College in Anya's lab, and I'm involved with some of the development for sort of core Symbulation functionality and default mode.
Yeah, hi, I was at Carleton College and I graduated this year and I'm starting a PhD at University of Vermont next year. And I was in Anya's lab at Carleton and also did similar research with Symbulation.
Awesome. And then Allison Cameron will be joining us a little bit later, helping with things also from my lab, also graduated last year and worked with one of the particular modes focused on lysis lysogeny. So that will make sense in a little bit. All right, so with that, okay, so as I said, starting to get things installed. The main things, if you're on Windows, Symbulation doesn't run on Windows, so we need a Linux environment, so Windows subsystem for Linux WSL. That's probably going to take a while to download and get set up. So if you don't already have that set up, we have an alternative option. And then we need to use a particular compiler and a nice Python library called Cookie Cutter that makes it easy to kind of set up a project. So links for downloading those things are on the tutorial page, and those are the things to be starting downloading now if you don't have them. If you can't install or you run out of time because there's a lot of things that you would need to install, that's fine. I made a link to a platform called Replit that basically will run this in a browser. So I would just recommend following that link and making an account so that then you can use Replit instead. There's a couple of things that won't quite work, but you'll be able to do the majority of things. Okay, so while things are installing, I am gonna kind of give you a conceptual overview of Symbulation. So very high level, it's an agent-based modeling platform. So we have populations of agents, little, individual objects that are going or that can do things. And it's specifically focused on the evolution of parasitic to mutualistic symbiosis. So there's lots of agent-based modeling platforms out there. I've tried to study parasitism, mutualism in most of them, and most of them don't work very well for this. So this platform is specifically for kind of studying this particular dynamic. And mechanically what that looks like, and like things that might be relevant to whether you want to use it. It's open source on GitHub under the MIT license. It's written in C++, so this lets us, optimize fairly well. But it does mean that there's a little bit of a learning curve to be able to use it, or to not to use it, but to make changes to it. And then there's two main versions that you'll interact with. So there's two main ways of interacting with it. So there's command line without a graphical user interface, and that's kind of the way that you would generally run most experiments and do kind of your main things. And then we actually have a web GUI, a browser-based web version that has a graphical user interface that's built on top of the research software. So these aren't two main versions, as kind of a misnomer. They're the same version, but two different interfaces, which we think is pretty cool. So the kind of hands-on components will walk you through those two different ways of interacting with it. It uses a platform called Empirical, and that uses Emscripten for making the web version. So this is what allows us to have C++ at the core to then cross-compile to a WebAssembly version of the software, which is pretty cool. And then there's kind of some core principles that we are following with this that is just kind of the philosophy that we're using. So our goal is to be modular. So with this kind of software, right, there's always like, oh, like we'd want to try like this way of doing it and this way of doing it. And we think all of those versions are great. And so we want to enable all of them to exist while not kind of interfering with each other and getting super bloated. So we are kind of taking a modular approach. We have a bunch of different modes for counting, and those modes aim to reuse as much code as possible between them, while also letting them be independent, so you can make extensions without messing with other people's kind of way of doing something. This is true of kind of empirical based things in general, but we push as much computation as possible to compile time so that runtime is faster. What this means is that it takes a while to it takes like a noticeable number of seconds to compile. And so don't be surprised when you compile it that it's, actually taking a bit of time. That is what enables us to then run experiments more quickly. So that's kind of one of our core goals. And then a lot of scientific software, we aim to have a thorough automated test suite. So we have a pretty good test coverage, if that's a buzzword that you're aware of, and documentation. So we're very much aiming for this to be a platform that you feel like you can trust, because it is well tested, so that things don't suddenly break, and it's well documented. So you feel like you are able to kind of understand what all the different tools available are, and can figure out ones to use. So with that, what does it actually do? So I'm going to be focusing on the default mode. So this is kind of, if you've seen any of my previous work on Symbulation from like more than just this ALife's presentations, default mode is probably what you're already familiar with, that we do have some cool new features. But like I said, there's other modes as well, depending on what you're interested in. So in default mode, we have, first we'll focus on just an individual kind of cell in the world. So each space in the world can have a host. Each host can have 0 to more endosymbionts. And they can have this interaction value that goes between -1 and 1 for antagonistic behavior to mutualistic behavior. And so this is like, in this scenario, the host is kind of neutral or maybe even a little bit mutualistic, and the endosymbiont is stealing some resources from the host. But then this could also be a mutualistic relationship where the endosymbiont is donating resources to the host. In addition, there can be an ectosymbiont in a space. And currently we only have support for one in a cell, but that is something that we definitely could branch out in the future. So this is a symbiont that doesn't live inside of the host, but can still interact with the host. So if we then zoom out, no, that's not the direction I want to go. There we go. Okay, if we zoom out, we then have a population of these different hosts and their endosymbionts, and then their various ectosymbionts that are all kind of interacting in a world together. And what that actually looks like in the code is that there are two different populations in the actual code. There's a host population that has the hosts and their endosymbionts, and then there's an ectosymbiont population. And this is how we make sure that the ectosymbionts aren't competing with the host for space, right? They're able to be in the same kind of cell as a host without like excluding each other. So if you're looking through the code and you see that there's these two different populations, that's what's going on there. Okay, I'm going really fast through this because there'll be time for questions, and I want to make sure that we get to the hands-on portion. So hold questions or post them in the chat and people can, everyone else can answer them as well. So if you are wanting to use Symbulation out of the box and make changes to it through our existing configuration options, there's a whole lot of ways that you can do that. So we have many different configuration options and they can be changed at runtime, which is very nice. And they all live in this simsettings.cfg file. So once you've gotten your project downloaded in the hands-on, you'll look to the end of the files to find that. There's a number of different sections, and the ones that are probably going to be most relevant to you today are the main group values, and then mutation if you're interested in messing with the mutations. There's others that are relevant to the other modes that you're welcome to look at and mess around with, but they probably are going to be less interesting for you to try out today. So a couple basic ones that the hands-on guide walks you through are vertical transmission. So this is the probability that a symbiont gets copied into the host's offspring when a host reproduces. And generally what we found in the past and what's been verified with other systems as well is that if we have a higher vertical transmission probability, then we expect more mutualism to evolve because the symbiont has a selective advantage if it helps the host reproduce because it benefits from that. And then also in particular, grid X and grid Y control the dimensions of the world. And so when you start running an experiment, if it's running pretty slowly and you'd like to speed it up, making those smaller will be kind of the most effective way of speeding things up. Obviously, if you were trying to run an experiment for actual data that you wanted, you wouldn't want to make that too small. But today, when you're just trying to kind of mess around, making those smaller is a good way to get results a little bit faster. I'm going to quickly show you what it looks like to actually run the software, or showing you an example. Again, this is all in the hands-on guide that you'll walk through. I just want to give you a preview. So once you've built the simulation software, you would run it with /simulation_default. And then here's that vertical transmission setting that I told you about. So here we're setting it to 10%. So that's a very low chance of the symbiont getting copied into the host's offspring. It's always good to specify a file name so that you have, different treatments have different names. You don't accidentally overwrite things. And then here I set the grid to X to 50 and the grid Y to 50, just to make it a little smaller, a little faster to run. So symvals, and then whatever your file name is going to be the kind of default data that's printed out. So when you run this, you'll see that there's this data file that's created. And there's a lot in here. These column headings are semi-descriptive. What you're seeing here is the mean int val. So that's the mean interaction value, that value that determines where they are on the antagonism to mutualism spectrum. We see here that the average value is -0.5, so that's pretty parasitic. So we're seeing already after just a couple hundred updates that at a low vertical transmission, we're getting a parasitic average. And another cool thing that you see in this file is this distribution. So we actually frequently see stable coexistence between different populations. And so this is histogram bins. So these are bins. So this is from -1 to -0.9, and then every 0.1 up to 0.9 to 1. And then you see here that we have a distribution of different values, but they're pretty skewed towards the lower end. If we then instead set the vertical transmission rate to 70%, so here it's the same command, except we've changed the vertical transmission to 0.7. And so then I also changed the file name so that we don't overwrite our previously existing file. And here we see the mean int val is up higher, 0.7. So we have a nice mutualistic average there. And we see here the distribution collapses towards the mutualistic end of the distribution pretty quickly. So different dynamics going on there too. We're not done. There's more settings to try. There's lots more settings to try. A couple of others that you might be interested in messing around with. Grid is whether there's spatial structure in the world or not. So by default, offspring are placed just randomly in the world. If you turn on grid, then offspring are placed next to their parent. This is true for both symbionts and hosts. So you could see what effect you see with that. You can turn on the free living. So by default, those ectosymbionts aren't actually allowed to exist since previous versions of the software didn't have them, but you can turn those on to allow symbionts to live outside of the host and see what that does. And then if you are interested in changing like how quickly evolution proceeds, you can change the mutation size to make mutations larger or smaller on average. Like I said, there's a lot more settings, but those are just some ones that might be interesting to try out. Okay, so with all of that, the next part of our program is the first hands-on. So the first hands-on walks you through cloning down the cookie cutter project, building and running it. So just getting the kind of basic stuff up and running, building and running the web GUI version. If you're interested in doing that, there's a guide for that. And then running a few replicates, tweaking one of these values, and then seeing what results you get and trying it out. My goal for this is that I make sure that you have enough time to actually do at least one of these things. And so we're going to structure this. We're going to open up breakout rooms. We're going to have different breakout rooms for each of these guides. So you can talk to other people that are working on the same one, and we'll distribute ourselves to the different rooms to help and answer questions. So room 1 will be building and running. Room 2 will be building the web GUI. And then room 3 will be collecting and analyzing data. Obviously, you need to build and run before you can collect and analyze. So feel free to change rooms too. And then the main room will be for general questions, since I haven't given you a chance to ask questions yet, and anything else that you want to do. Okay, how does that sound for a plan? Good. Okay, I have the power to open breakout. Okay, our recording is back. That's exciting. Okay, so I think we will start again, and hopefully we don't get we don't lose people. Okay, thank you everyone. I hope that you had a restful break. We didn't have any extensions, ideas, so that's fine. We'll pivot from that. Before we jump into talking about the code structure more in depth, was there questions that you thought of during break that you'd like to ask? Okay, cool. Yeah. So for the code structure, I'm going to talk about kind of very quickly how the code is organized. And give like an overview of what the guides kind of walk you through more step by step. Like I said, as before, there's guides that kind of detail, go through different parts. So I'm going to go quickly so that then we have enough time for the hands-on components. And so with all of this, I feel like it's better to like actually try and do stuff instead of just listening to me the whole time. Okay, so I need to move my participant screen because it is blocking my view, despite how lovely all of you are. Okay, so the code of Symbulation is organized into three main classes that kind of are the powerhouse of the functionality. The first is SymWorld. So this is the environment that holds all of the organisms and is doing the majority of the heavy lifting when it comes to kind of making sure that everything's running. So it has a variable pop that is the host population, and then a variable sympop that is the symbiont population. It inherits from Empirical's world class, so it has a lot of functionality that is not specified in the SymWorld file, including pop. Like pop isn't specified in the file, it's used by the file because that's from Empirical. And then it has a whole bunch of methods, but the two that will be like most relevant to you are run experiment. So that's what actually, you know, has like all the pieces run. And a lot of what run experiment does is call update the correct number of times, and update is what happens in a given time step. So it goes through all of the hosts in the world and lets them do their thing. It goes through all of the symbionts, the free living symbionts, lets them do their thing. And it's also where like resources would be adjusted or like any of that sort of stuff that you want to have happen on every update. The next kind of core class, as you might suspect, is the host. So the host class has, again, a bunch of stuff, but the core things are the interaction values. So that's the value between -1 and 1 that determines how it acts towards its symbionts. So that's its genome. It has a vector of symbionts called Syms. So the host, those are its endosymbionts that it allows to do whatever. And then it has an instance variable points that are the resources that it needs to collect that then when it has enough, it's able to reproduce. And then its kind of core method is process. And so process is what's called by update. The host is in charge of doing whatever it wants to do in a given update, and that's all specified in process. So if you want to change how a host acts in a given time step, process would be the method that you would look to do that. And then hosts, as I said, have their vector of symbionts. So the other core class is the symbiont class. The symbiont class, again, has an interaction value. That is its genome. It's how it interacts with its symbiont or with its host, whether it's -1 highly antagonistic or +1 highly mutualistic or anywhere in between. It has a instance variable myHost, which is a pointer to its host so that it's able to kind of call host methods and keep things organized that way. It also has an instance variable points, since it also has to accrue resources in order to be able to reproduce. And then it also has a process method. And this is what either the host lets its endosymbionts do actions by calling their process method, or the free living symbionts SymWorld calls it. So again, this is what they do on like a given time step. If you're interested in changing their behavior, that would be the place to do it. You might notice that there is a bit of a loop here where symbionts know about hosts and hosts also know about symbionts. And actually what is not specified here is that hosts and symbionts also both have pointers to their world. And so that leads to problems in C++. And so there is a superclass organism that is just doesn't actually do anything, but is what everything knows about, so that we don't have include cycles. So like C++ won't let you have one file include another file that then tries to include a file that it's dependent on. And so you'll see this organism superclass, and it's annoying, but it's the current way that we are getting around that problem. There are certainly other ways of getting around it, but it's a process. So if you see organism, that's what is going on there. It's just a class that doesn't actually do anything to let us get around that problem. Okay, so if you want to make a change to Symbulation, so The default mode, lets you do a lot of different stuff, but almost certainly there's some new thing that you want to do since you want to, be able to write a paper about some cool new result. There's two different ways to do that, one more involved than the other. So the first thing that you could do is add a configuration setting. So if you're just trying to do kind of a simple change that, is just a little tweak, configuration setting probably will do what you want. And the nice thing is that this, you can change at runtime through that configuration file. So this is the place to be able to, try out a number of different values for some setting to see what effect it has. Yeah, like I said, see effect of change easily via the command line arguments. And the really nice thing is that to do this, you only have to change 2 files. So I'll show you kind of what that could look like, but it's a pretty straightforward thing to do, which is nice. If you want to make more changes than that, then you would just probably enter into the territory of making a new mode. Making a new mode means making new subclasses of the existing classes. So this is, you're really wanting to overhaul, the genome and the functionality and everything of the host of Symbiont. So you're wanting to kind of really make a new thing. You also have to make a new compile target because you're going to be compiling a new version of the software. And so there's going to be a number of new files to create and change. So it's more involved. It requires, being pretty comfortable with C++. But, that's, something you might want to do. So I'm going to first focus on the configuration setting and walk through kind of an example. So the syntax, so the change that you're going to be making is in the Symbulation EMP source config setup.h. This is where all of the configuration settings are specified. And the syntax is pretty, hopefully not too bad. So value is always going to be there. And then name of setting is whatever you want your new name to be. We have the convention of having it be all caps. And then you need to specify what type the setting is going to be. So this is, Boolean or int or string or double. And then a default value. So if the user hasn't set it to something else, what it should be. And then just an English description that is kind of what you see in that sim-settings.cfg, the kind of string describing what it is. The really nice thing about this config setup that is all provided by Empirical is that getters and setters, accessor mutators are provided automatically. So once you've made your configuration setting in config setup.h, you will automatically be able to do whatever name of setting parentheses on the config object to get the value that setting is. And also you can set it just by putting something into the parentheses. That then is a setter to set the value. So that's nice. You don't have to go and make a bunch of new methods if you're adding new configuration settings. All of the core classes have access to a shared configuration object already called myconfig. So if you're in any one of those classes, you're able to just do myconfig arrow name of your setting in order parentheses in order to get the value. So that's why you don't have to actually make changes to very many files because a lot of that's provided automatically. So if we look at a specific example, we need to first add our setting to config setup.h. Then we need to go into whatever file we want to actually have that setting influence and make whatever logic we want to do. And we would use this myconfig name of setting to get the setting value and then do whatever logic we want. So for example, if when I implemented limited resources in the world, I went into config setup.h, I added this line value, limited res total, it's going to be an int. I could have made it a double, that would have been fine. We frequently, by convention, we use -1 to mean like this setting doesn't do anything. So here, right, -1 is unlimited. Otherwise, the number given is going to be the number of total resources available over the entire run. And so that's all I had to do in order to make the new setting. And so then in Sim World, I made a little instance variable to keep track of how many resources there were. I set that in the constructor from my config. So I just went and grabbed, whatever value is there, and I set it in equal to my instance variable. And then I already had this method called resources. And in that, I just put in a check. If the total resource is -1, then that means that it's supposed to be unlimited. So just give the organism whatever they wanted. Otherwise, do the logic for subtracting the desired resources from the total resources, or check to see if they're out and return that. So these are the only changes that it needs to make, just adding it to the config setup and then using it for the purpose that I want it to use. So that is, if you're wanting to make a small change and just kind of, tweak a little thing and uses however much kind of C++ you want to do, depending on how complicated your logic is. If you want to make more changes and you would need like a whole bunch of configuration settings, then it probably would make sense to do a mode instead. And so modes have their own subclasses of each of our core classes. You don't have to have a subclass if you don't want to, but you can. And then like I said, you need to make a new compile target. So you need to go and edit the make file to add a new target. And then they'll need to probably do their own data collection because there's probably new data that you want to collect. And then of course, anything else, any other changes that you want to make. So the modes are really the way to kind of open playground, do whatever you want. So I'm going to go through an example quickly. So to make a new mode, you would need to make a new folder, Symbulation EMP source, and then you make a folder, whatever you want, kind of a little short name of your mode to be. And then so for example, for a new mode that focused on a specific kind of relationship between bacteria and viruses that infect them. So we called it lysis mode. And we then, I say we, Allison, then made new host and symbiont subclasses. And these need to extend either the host or the symbiont or existing subclasses so that it follows the kind of class chain. And the way that you do that in C++ is just the name of your new class, colon, and then public, and then the name of your of the superclass, so in this case, host. And so this is in bacterium.h if you want to see an example. And then make a new world that extends sim world. So the example is lysis world.h, follows this same syntax. And then you need to make a new what's called world setup.cc. This file is currently needs to be mostly just copied from existing. We're working on improving the duplication of code so that there's less that needs to be copied, but it's a work in progress. And this is all detailed in that the guide. So this is not stuff that I expect you to memorize at this moment, just kind of going over the steps so that you know what you're looking for. And then you'll make a new native file. So this is the .cc that is what's actually run. And again, it's going to be the name, the short name of your mode. It's also mostly copied, though we're improving on that too. So example is Symbulation lysis.cc. And then you need to add a target to your make file to allow you to easily make it. So for lysis, we're calling it lysis mode. So this is like when you do make you do make lysis mode. And then the main, like this is going to look pretty much the same, except that you're going to change lysis there, and lysis there to be your own mode name. There's a whole bunch of flags that are needed in order to compile all of this. And so that's why we the make file is a little bit overwhelming, but you just have to look for the spot where the other modes are and make your own line. Okay, good. Since the keynote is in 10 minutes, I want to make sure that we are not late to that. So I think we will jump to wrap up debrief, but hopefully this gave you a very brief introduction to this, and maybe more importantly, an introduction to us. So You know that we're not scary and happy to talk, and I'm super happy to talk with anyone further about any of this and help. If there's stuff you're excited about, very, very happy to help and try stuff out. So I need to once again move the participant screen. There we go. Okay. I'm gonna close this. We have a surprisingly strong showing of Symbulation work at ALife this year, or maybe not surprisingly, I don't know. So this is at the end of the kind of tutorial main page. So again, nothing to memorize here. But if you're interested in how we are using this platform currently, so therefore other ways that you can do things with it, there are a bunch of talks. So yesterday, one of my students, Krish Gardis, presented on the efficient mode, looking at another thing that can select for mutualism, and that recording is up. So it's on in the Slack channel under the recordings. That's on the Monday last afternoon slot. And then tomorrow, Kira Piper, Emily and I are presenting on the active symbiosis, endosymbiosis new feature, and then Allison and others are presenting on the lysis mode stuff. And then Thursday, I'm giving a kind of brief overview of symbiosis and digital evolution in general. And it's one of those extended abstracts, so it's mostly just advertising for a paper that's already published. I'm kind of doing a very large overview of all of these different things that people have done. And then Emily and I and some students are presenting on multi-infection. So when you allow for more than one symbiont in a host on Friday. So yes, lots of different places to see cool things that people have done with Symbulation. That feels very self-indulgent to say since I am on all of these papers, but I think it's cool what my students and colleagues have done. So with the 8 minutes we have, I would really be interested to hear feedback or thoughts or whatever on if this is something that you could see using in research or teaching. And what additions, right? If you're not interested in making additions to yourself, but there are additions that would make it useful to you, would be very, very happy to hear that. And then thank you so much for coming to this tutorial. It's the first time that I've run something like this. And it was very good motivation to get all those guides written. But I appreciate your time very much. And I will be probably sending out a survey with similar questions like this in case you're not feeling like answering right now. But since I really would like feedback. So we have about 7 minutes before the keynote. I totally understand if you need to take a break before the keynote, but if you feel like sticking around and have things to say, we'd love to chat. Thanks so much.